Hey everyone, and welcome. Today, we're going to crack open the instruction manual of life itself. We're going to explore this absolutely fundamental tool that scientists use to read, compare, and, understand the genetic code that makes every single living thing on this planet unique. So let's just dive right in and decode the hidden tool that's changing our world. So here's a question you've probably asked yourself. Back in 2020, the entire world was suddenly up against this new, completely unknown virus. And yet somehow, in less than a year, we had multiple working, effective vaccines. I mean, how on earth was that even possible? How did researchers move so incredibly fast? Well, a huge part of the answer comes down to this really powerful computational method, sort of a hidden tool that's working behind the scenes in labs all across the globe. It's a technique that lets scientists make sense of just massive, overwhelming amounts of genetic data and do it at lightning speed. And here it is. It's called sequence alignment. By comparing the genetic sequence of the new virus to others we already knew about, scientists could zero in on its most critical part, that infamous spike protein. And that was the key. That's what unlocked the door for developing the vaccines from Pfizer and Moderna. It really did all start with alignment. Okay, so what is this thing, this revolutionary technique? How does it actually work? Let's break down the core idea piece by piece.
At its heart, sequence alignment is really just a way of arranging those long strings of genetic letters, the A's, T's, C's, and G's of DNA, to find places that look alike. And these similarities, they aren't just random. No way. They're actually clues, little breadcrumbs that can tell us about what a gene does, its 3D structure, or even its deep, deep evolutionary history.
Honestly, the simplest way to think about it is like a super advanced spot the difference puzzle. But instead of comparing two cartoon pictures, scientists are comparing these incredibly long strings of genetic letters. And they're looking for matches, for differences, and even for gaps. And all of that tells a biological story. Right, so it's not like there's just one button scientists push. It's more like they have this whole toolkit, and they have to pick exactly the right alignment tool for the specific question they're trying to answer. The two main players here are pairwise alignment, which is when you compare just two sequences. That's perfect for, say, seeing the difference between a healthy gene and a mutant one. And then you've got multiple alignment, which compares a whole group of 3 or more at once. That's great for studying an entire family of related proteins. Now, to really get into the nuts and bolts, we're going to focus on the simplest type, pairwise alignment. And even when we zoom in on just pairwise alignment, there are two major strategies, and each one is asking a totally different kind of question. We call them global alignment and local alignment. And this is the absolute key difference, right? A global alignment asks, okay, are these two sequences similar from start to finish? It tries to match them up across their entire length. But a local alignment asks something much more specific. It asks, hey, do these two sequences share a small, really similar region, even if the rest of them is totally different? It's all about finding that one golden nugget of similarity. All right, let's tackle global alignment first. Remember, its whole goal is to find the best possible match across the entire length of two sequences. It uses this really clever method, often called dynamic programming, which is basically just a fancy way of saying it breaks a giant problem down into tiny, manageable pieces to find the absolute best answer. It might sound a little intimidating, but you can really boil it down to 3 main steps. First, the algorithm makes a grid, kind of like a spreadsheet, with one sequence along the top and the other down the side. Second, it goes through every single box in that grid, giving out points for a match and taking away points for a mismatch or a gap. And finally, it traces backward to find the path with the highest score, kind of like finding the best route on a map. And this is so cool because you can actually see that process right here. This whole grid, that's the scoring matrix. Each number is the best possible score at that position. And that red line snaking through it, that's the traceback. That is the optimal path that the algorithm found, going from the bottom right all the way back up to the top left. And check this out. That path through the grid isn't just some random line. It's basically a set of instructions. Every time it takes a diagonal step, that's a match. And every time it goes straight up or across, it tells the algorithm to insert a little gap, which you see here as a dash, showing where something was added or deleted in one of the sequences. Okay, but global alignment has its limits. It kind of assumes the two sequences are pretty similar overall. But what if they're not? What if you're comparing two really long, mostly different proteins that just happen to share one tiny critical piece? For that job, you need a different tool. You need local alignment. So local alignment uses a similar grid, but there's a game-changing twist. Instead of starting at the bottom right corner, it hunts for the one cell with the highest score anywhere in the whole grid, and it starts there. Then, it traces back only until the score drops to 0, and then it just stops. And look at what you get. The result is fundamentally different. It doesn't give you an alignment of the whole sequence. Nope. Instead, it just pulls out that one single best region of similarity, that highest scoring local path. It completely ignores all the surrounding stuff that doesn't match up well.
Okay, so we've gone through the how, the matrices, the tracebacks, all the algorithms. But let's circle back to the biggest question. Why should we care? Why does any of this really matter?
The power of sequence alignment is it's immense. We use it to build evolutionary trees to see exactly how we're related to chimps or even to bananas. In medicine, it's like our magnifying glass for finding those tiny genetic typos that can lead to disease. And when scientists find a brand new gene, alignment is the very first tool they use to ask, okay, what does this thing do? It's the foundation for mapping entire genomes, and, as we saw with COVID-19, for designing life-saving drugs from the ground up.
It's pretty wild, right? It all just boils down to this one powerful idea, comparing strings of letters. It is, without a doubt, a cornerstone of modern biology.
So from tracing our ancient past to designing the medicines that stop a pandemic in its tracks, sequence alignment is our Rosetta Stone for the language of life. And as our ability to read that genetic code just gets faster and cheaper, it leaves you with a pretty amazing thought. Our own genetic code is billions of letters long. What secrets are still in there, just waiting to be read?